It is well known that in Alzheimer's disease, one of the earliest events that is pathophysiologically relevant is the loss of synapses, and that correlates best with cognitive decline. It is also well known that a large number of genes are associated with a risk of Alzheimer's disease, and specifically variations in a large number of genes are associated with increased risk for Alzheimer's disease. But the biology of these genes is largely unknown. The work that I described today, the work that I described in my talk was part of a larger set of studies that utilizes human neurons, which we derive from stem cells, iPS cells and ES cells, to interrogate the functions of the genes that are associated with Alzheimer's disease and to understand better what happens when these genes are mutated, as is observed in some cases of Alzheimer's disease. The critical component of these studies that I discussed is that we are not just mutating these genes and clones that we then propagate and use to make neurons. Instead, we are generating mutations that are conditional and thereby are able to analyze neurons that are precisely isogenic. Because one of the problems in studying human neurons that are derived from iPS or ES cells is that if you study mutants, they are rarely isogenic, truly isogenic, because they're always derived by clonal propagation of mutated cell clones, which are no longer isogenic, even though in the literature they are called isogenic. This allows us to analyze precisely the effect of genetic variation on neuronal properties. It should be noted these are exclusively studies of neurons and not of astroglia, not of microglia. And by no means do these studies imply that astroglia or microglia doesn't have a major role. It does. It's just that the studies focus on neurons because neurons are, after all, the cells that form the synapses that are impaired in Alzheimer's disease. And so that's why we study them. And our studies, in a nutshell, show that somewhat counterintuitively, genetic variants associated with an increased risk in Alzheimer's disease in human neurons confer an increased ability to form synapses, not a decreased ability. A finding that has either perplexed my friends and colleagues or made them feel slightly cautious. We can't explain these results in the sense of the disease, but we also feel that it is a little bit naive to think that these gene changes would necessarily be detrimental to synaptic function itself. Because after all, Alzheimer's disease is a disease that develops over decades, not something that is apparent in childhood, as you would expect for any genetic change that impairs synaptic function directly.